Plastic is everywhere. It's in the food we eat, the water we drink, and also in the air that we breathe. So what does this mean for our health? To date, there is a lack of scientific evidence on the effects of ingesting plastic, and only a few studies have attempted to estimate how much we're actually ingesting. So my study has addressed this research gap by investigating the microplastic exposure to humans from shellfish consumption from different regions of the world. My name is Lexi Crouch, and the title of my research project is From Ocean to Plate, Microplastics in Marine Bivalves. So today I just want to provide a bit of background information about my research project and the motivations behind my research. I will discuss the research problem and the objectives, the specific objectives born out of that. I'll discuss the methodology that I use to achieve these research objectives and the findings from the literature review. Finally, I'll give an overview of the results from my research and their implications for what they mean for human health, bivalves and aquaculture industries. And I'll finish up with some concluding remarks and recommendations for further research. So when plastic enters the ocean, it doesn't break down. It breaks up into smaller and smaller plastic pieces called microplastics. And microplastics are everywhere in the ocean. They've been found on the ocean surface, all in the water column, and even in the sediments. Several research studies have found marine organisms to ingest these microplastic particles, including fish, crustaceans, and bivalves. So marine bivalves including species of mussel, oyster, clam, and scallop, are particularly vulnerable to microplastic ingestion because they filter the surrounding seawater, meaning they accumulate microplastics as they feed. Now, microplastics are defined as particles that are less than 5 millimetres in size, and they can occur from the fragmentation or the degradation of larger plastic items that are in the ocean, but they can also be manufactured deliberately, such as the microbeads that are found in personal care products and cosmetics. So back to bivalves. Several experiments have actually observed the harmful effects of microplastics once they're ingested by different bivalve species. Some of these effects include accumulation in the digestive gland and in the gills of bivalve species, neurotoxicity, it's impacted DNA and caused oxidative damage as well. It's also been demonstrated in laboratory experiments to cause delayed larval development, interference with energy uptake, and reproduction issues as well. So my marine bivalve species, many of them, are commercially important species. They're not only valued for their edible meat, but also for their shells and the pearls from some oysters. They also have ecological significance, and many species are considered keystone species. This is because they provide ecosystem services such as carbon sequestration, they cycle nutrients in the water, provide coastal protection for our coasts, and they also serve as a habitat for other species and assemblages in the environment. So bivalves in particular pose a concern for human health and exposure to microplastics because, unlike other seafood items such as fish, bivalves like mussels and oysters are consumed whole with the gastrointestinal tract intact. So this graph that I developed kind of explains how microplastics are going from the ocean to our plate. So you can see here that when bivalves are cultured, they're cultured and produced in the natural marine environment. And as I mentioned earlier, microplastics are present in all marine ecosystems. So then bivalves filter these microplastics in the water along with their natural nutrients and their food. After after they're harvested, bivalves are often depurated in fresh seawater. During this process, some microplastics are ingested during depuration, but some remain. And this leads to the final step where they end up on our plate and they're ingested by humans. So this study aims to investigate the regional differences in the uptake of bivalve species, but also looking at the subsequent dietary exposure to shellfish consumers across the globe. So this overarching research problem was framed into 4 research objectives. The first objective is to identify and synthesize data to produce robust estimates for microplastic ingestion by bivalves in different regions. Objective number 2 was to look at those regional differences in microplastic ingestion by bivalves. Objective number 3 was to look at the human dietary exposure. So how much are people eating in different regions of the world as a result of shellfish consumption. And the final research objective was looking at synthesising the literature and the current best evidence to understand what the risks are to human health based on the findings of Objective 1, 2 and 3. It's important to mention the scope of this research quickly as well. So this research focused on filter feeding extant marine bivalve species, the ones that are consumed by humans. So species that aren't consumed by humans are excluded from the study because this study is all about the impacts on human health and exposure to humans. Freshwater species and deposit feeding bivalves are also excluded as less public research is available on them, and this study, the research problem, is focused on microplastics in marine environments. Moving on to the methodology. So this study, it didn't use field work, it actually focused around secondary data analysis and empirical desktop research, and it used this information and data available in the literature to extrapolate, estimate and calculate microplastic uptake for bivalves in some regions and also for human dietary exposure in different regions, both for the present and for the year 2100 to see what the projections are likely to be if microplastic contamination of our marine environments continues. So the research methodology consisted of 4 phases. The first phase involved systematically reviewing the literature to build a database of microplastic loads from different countries. Now, it's important to note that different studies use different analytical and laboratory procedures and techniques to quantify microplastic uptake or body burden in bivalves. So this made comparison really difficult. So what I did is developed a systematic selection process based on a set criteria for selecting the best studies and the most comparable data to include in my database. So this systematic selection process was based on the analytical technique, the laboratory procedures used, the reporting units reported, and the most recent research as well, some recency. You can refer to my dissertation for some more information about this process, the selection process, how the metadata was extracted and aggregated. So phase 2 involved estimating microplastic uptake. So from the literature review, it was evident that studies on microplastic occurrence in bivalves were missing from many regions. So the literature review identified 20 countries that had reported microplastic body burden in bivalves. So I developed a simplistic model to estimate microplastic body burden loads in bivalves from 11 other countries. So this model, referred to as the bivalve lifespan uptake model, or the BLUE model for short, uses key data from the literature to estimate microplastic uptake over the lifespan of a Mytilus edulis mussel species. So the model uses microplastic surface water concentration as well as the filtration rate ingestion rate and depuration rate of this species of mussel. The model is really based on the positive correlation between microplastic concentrations in surface waters and microplastic contamination in bivalves in those waters. So this is explained in more detail in my dissertation, but essentially there was a positive correlation between those two variables, and it was demonstrated by numerous studies, including Kazor and others, that was in 2019, Lee and others in 2019, and Q and others in 2018. So it's been recently, this correlation has been recently proven in the wild and in laboratory conditions as well. So this diagram provides a bit of an overview of the blue model. So essentially an 18-month lifespan was estimated based on the lifespan of mature mussels cultured in aquaculture systems, and a study had demonstrated how this mussel filters water that is contaminated with microplastics. So this was used and extrapolated out to 18 months, and the mussels filter approximately 18,000 litres of water over that lifespan. So then, of course, we've established that microplastics are in the seawater. So Woods and others, 2018, did laboratory studies that showed Bacillus edulis to reject 71% of microplastics that it filtered. It ingested approximately 9%, and it egested less than 1%. So this ingestion rate of 9% was used to see how much it would actually be ingesting over its lifetime. Then depuration, as we mentioned before. A study estimated the effectiveness of depuration over a period of 48 hours and found that 19.1 of microplastics still remained within the organism after the 48-hour depuration period. So this 19.1% retention rate was used, discarding 80.9% that were assumed lost during depuration, and that came up with a figure of how much would be in the bivalve available at the time of consumption. So robust and conservative assumptions were made to estimate microplastic uptake, and this is explained more in my dissertation. To take this to the next level, I used projections by Everett et al. And in 2019, who estimated the number of microplastics that will be floating in the oceans in the year 2100. And I used that to extrapolate to the body burden of bivalves in all these different countries using the statistics from the literature and the blue model to estimate potential body loads. Phase 3. So phase 3 involved the calculating human dietary exposure to microplastics. So this was estimated using average shellfish consumption data for each region. Microplastic exposure was calculated for shellfish consumers in 31 different countries in terms of number of particle, but also in terms of mass. And the calculations and assumptions used to calculate this mass are described in detail in the methodology section of my dissertation. So only 3 other studies have actually estimated dietary exposure to shellfish consumers. So my methodology used the same methodology as these practitioners, which again is outlined in my dissertation. So as you can see here, for Van Korlenberg and Janssen in 2014, estimated a consumption rate for European top consumers to ingest about 11,000 microplastic particles per person per year. In Korea, it was a bit lower, 212 microplastics per person per year, and in Tunisia, it was lower again, 23 to 44, based on the low shellfish consumption rates in that area. Phase 4 synthesised and discussed the best literature, the best available peer-reviewed literature on the impacts of microplastic ingestion on human health to try and understand what the potential risks are. And these were discussed in the context of the results from phase 1, phase 2 and phase 3 of the methodology just outlined. And we also looked at what human dietary exposure would look like in the year 2100 as well. Research outcomes. So looking at objective number 1 and number 2, which looked at microplastic contamination in bivalves in different regions. So microplastic uptake from 20 countries was obtained from the literature, and the blue model estimated uptake in an additional 11 countries, and the results are here highlighted blue. So results indicate that contamination is widespread in commercial bivalves across the globe, with microplastics detected in every single country except one, which was Denmark. So microplastic contamination in bivalves is highest in Russia, South Africa, the Netherlands, Indonesia, and Italy. And it's lowest in Mexico and Australia. And of course Denmark, where microplastics were not detected in bivalves. So when we look at these results, we can see that Russia and South Africa are quite high, and this again is because the model relied heavily on microplastic contaminations of surface waters in those regions, and the locations where these surface waters were sampled were really contaminated areas. They were enclosed bays and harbours, which lots of industrial outputs going to those regions. And the contamination levels recorded in those bays were actually not the highest at all in the literature. This suggests that the results are reasonable and that the rates could be higher in other regions where microplastic contamination of surface waters is a lot higher. So objective 3 was about estimating dietary exposure to shellfish consumers across the world. So dietary exposure to consumers was estimated, and the annual dietary intake of microplastics was highest in Russia. Almost 15,000 microplastic particles are consumed per year, per person in Russia. This is followed by South Africa, where it's almost 13,000 microplastic particles, Italy, where it's almost 12,000 microplastic particles, and China, 9,500 microplastics consumed per capita per annum. So based on the results of the 31 countries, I was able to estimate what the average shellfish consumer across the globe would be ingesting. And that worked out to be 7,700 microplastic particles per year from the global average diet of a shellfish consumer. So this also translated to the equivalent of 12,330 micrograms of microplastic. And then I mapped the results of all the countries so we could determine the regional differences across the world of geographic exposure. So you can see they're highest in Russia and South Africa, Canada also quite high, and then lower rates in some of the European countries and Australia as well. And again, low in Australia because of the low microplastic contamination found in the waters sampled in Australia. So looking at the results for the year 2100 has some interesting findings. So microplastics floating in the ocean are projected to increase 50-fold by the year 2100. So based on these predictions, the average shellfish consumer could be ingesting 360,000 microplastic particles per person per annum in the year 2100. I also took this a step further using the data for Russia to estimate the worst case scenario. So the worst case would be the average person would be ingesting 700,000 microplastic particles per year from shellfish alone. And this worst case scenario is the equivalent of 1.12 grams of plastic, which is the equivalent of a paperclip or a pen cap. So only one other study has actually estimated exposure levels for the year 2100. So that was Van Korwenberg and Janssen, again in 2014, estimated European shellfish consumers to ingest between 66,000 and 440,000 microplastic particles per year through shellfish, and that was for minor and top shellfish consumers. So the average projection that I estimated fell within this range, and of course Russia exceeded this range, so quite close results there when looking at projections for the year 2100. Objective 4 was synthesising the current best evidence from the literature regarding the impact of microplastic ingestion on us, on human health. So the potential risk to human health could arise from 3 avenues. One is physical effects of microplastics in the body, and this includes the translocation of tiny microplastic particles outside of the gut wall and into our cells and across the blood barrier and a number of other locations in our body. The second avenue is the leaching of chemicals that are actually added to plastics during manufacture. So these can leach out. And number 3 is the release of heavy metals and persistent organic pollutants that have actually sorbed from the surrounding ambient seawater onto the surface of microplastics. And because many of these chemicals found within and absorbed to microplastics are actually known carcinogens, endocrine disruptors and mutagens. So studies have shown that these toxins and particles can transfer up the food web to high trophic levels, and they can bioaccumulate and biomagnify in the tissues at every single level. And of course, at the top of the food chain is humans. So this is why there's such a concern for the potential risk to human health. So the effects of microplastics on human health is still an emerging field of research. And overall, there's currently a lack of conclusive evidence on the fate of microplastics and the risk to human health from either translocation or the accumulation of toxins as well. So in conclusion, bivalves are consumed whole and therefore they are considered a key pathway for microplastics to enter the human body. This study estimated dietary exposure to shellfish consumers for 31 different countries. This represents the most comprehensive investigation into the ingestion of microplastics by shellfish consumers to date. Results indicate that microplastic contamination is widespread in commercial bivalve species across the globe. And this is because microplastics were detected in bivalves from every country except one, which was Denmark. This study provides a start for predicting the risk of exposure to microplastics for bivalves, but also to humans. Because at present, scientists don't fully understand the risk that microplastic pose to human health. So this study will contribute to knowledge advancement and will assist researchers in understanding those risks to human health, with the goal of informing mitigation and management strategies to manage how much is ending up in our diet. So based on the results of this study, it is clear that individuals, researchers, industry and governments need to consider strategies to mitigate and stop further pollution of marine ecosystems by plastic debris, but also effects on our biota and human health. So to finish, I'd just like to talk about some recommendations for further research. So the results of this study could inform further research into the potential effects on bivalves at a population level based on the projected increases for the year 2100, because such increases could have significant implications for bivalve production, bivalve health, and flow-on effects to fisheries and aquacultures that rely on bivalve production, but also the people that are employed by these industries and rely on it for their livelihoods or for their key intake of protein as well. So to mitigate and manage dietary exposure to humans, guidelines for shellfish handling, preparation, depuration should be improved and optimised to reduce how much is ending up on the consumer's plate. Standardised methods are really needed to quantify and monitor microplastics in bivalves and marine ecosystems to be able to make more precise quantifications and easier comparability across studies. And to begin to understand what the risks are to human health, a greater understanding of the contamination of the entire human diet is needed, not just seafood. So this includes all food, drinks, and even airborne plastics. And factors such as the food preparation and processing should also be considered in this risk assessment. Finally, please refer to my dissertation for a full explanation of the research background, methodology, assumptions that were used in the Blue model and estimations, for the full results, the implications and what it could mean for bivalve health, human health, and impacts on industry as well, conclusions and recommendations for further research. Thank you very much.